Thanks a lot, Jennifer, for inviting me and for this opportunity to talk to so many experts on paper mills and research fraud. So I'm going to talk indeed about paper mills in the landscape of biomedical research. As you can see here, I have to start by saying that I am not at all a paper mill expert or an expert in research fraud. I am, as Jennifer said, interested in our research group in visualizing different kinds of data. Usually we work with biological data, single cell transcriptomic data, and so on. I'm not going to talk about that. And one of the topics, recurring topics in the research in our team is to visualize the complex data sets in 2 dimensions so that we can do exploratory analysis, look for weird things in a data set, and we're interested in large data sets and challenges that come in different data modalities, visualizing images, visualizing texts, visualizing different kinds of stuff. So from that perspective, we got interested in this, and this is the team, we got interested in visualizing the biomedical literature. I'll explain in a second what this means. And the stuff that I'm going to present is available as a preprint led by Rita, a PhD student here in Tübingen, who I think should be present tonight also. So everything is Rita's work. There is other people you can see here that were involved. So this is a preprint available on bioRxiv since a while now. It just very recently we learned that it's accepted to publication in a new journal called Patterns from Cell Press. So it will appear there shortly, hopefully within weeks. But that's what it's about. So we wanted to visualize the PubMed. So I think everybody knows the PubMed is the data, is the library, online library that one can query that aims to comprise the entire biomedical literature. So everything that is published on biology and medicine is indexed by PubMed. And they say here on the website that they have 35 million citations in there. So if you actually look at these 35, and the good thing about PubMed is that you can download all the data that they collect. So this is easy. You don't need to ask for permission. You can just download the entire PubMed and then analyze it on your computer. So we downloaded these 35 million citations, sorted out only English abstracts. We did this already 2 years back, or we started working on that 2 years ago. So in this published version, we're working with the data set that goes until the end of 2021. I'll come back to that later. And we only take English language abstracts and we sort out, we remove abstracts that are maybe empty or very short or somehow broken, or we consider them broken. So we do some initial filtering and we're left with 21 million English abstracts. So just to make it a bit more concrete, I took one of the abstracts from PubMed just to illustrate this. So this is a paper about paper mills, co-authored by Jennifer here, and here's an abstract. So this is pretty typical. An abstract is typically a paragraph around that long, so it contains maybe up to 10 sentences or so in English. So this is the data set that we're working with. We have this 21 million of these short texts. We're not using any other information. We're not using the titles, the authors, the affiliations, anything to construct our visualization. We only use these texts. So we have 21 million texts. How do we even begin to analyze that? We need to convert texts to some numeric representation that we can then work with. And well, we use a language model, a large language model, or a rather small large language model that we didn't train, that was pre-trained before by another team, that actually was pre-trained specifically on medical texts. So it's supposed to be good for representing this kind of data. And if you take this model, which is a neural network, the details of how it works are beyond the scope of my today's talk, and you feed this text into this network, then it does some processing and outputs a vector of 768 dimensions. So for this text, you get out 768 numbers, floating point numbers. And then we're feeding one after another 21 million abstracts through PubMed BERT. It takes a while, by the way. It takes, on a powerful computer that we have, it takes several days because the network is large and it takes time to do this processing. So out comes a gigantic matrix that has 21 million rows, if you will, and 768 columns. Now, this is now we have a numeric representation, and maybe I should say that the idea of why we're using a language model is that similar abstracts should give us similar vectors. So this model is supposed to know to give semantically meaningful representation of the texts. Let's say it like that. Okay, so the abstracts on the same topic should be mapped to 768 dimensional vectors that are close to each other in some sense. Okay, so we get this gigantic matrix that is still too large, way too large to visualize. We want a visualization, so we use another algorithm called TSNE. It's a method that exists since around 15 years to visualize any kinds of high-dimensional data. So high-dimensional now means 768, and we want a two-dimensional view of it. And the way TSNE works, which I'm not going to explain in detail, is that it looks for neighbors, so for vectors that are very close in this 700 dimensional space, and tries to arrange the points in 2D so that they also remain close together. So things that are like a tight cluster of similar papers should remain a tight cluster of similar papers in 2 dimensions. This also takes a while. So all of this, as you see, takes a while, but within several days we can run this pipeline, and we spent, or Rita, I should say, spent a lot of time optimizing different steps of this pipeline. What's the best language model to use? What's the best visualization algorithm and approach to use it so that it runs fast enough, and so on. All of that, I will not give you any more details about that, but we spent some time trying to make this work nice. So when all of that is said and done, we get an image like that. So what you see here is 21 million points in 2 dimensions. So you can't really see 21 million points, but if you imagine zooming in this figure indefinitely, you would eventually see 21 distinct points. We color them with several dozens colors that you see here, labels. These labels don't come from PubMed, but we use a trick to come up with these labels, and the trick is very simple. Everything that is published in the journals that are called the Journal of Gynecology, or the Annals of Gynecology, or the International Conference on Gynecology, everything that is published in venues like that gets a label gynecology, and so on, with all these different labels here. Okay, so stuff that is published in a journal called Nature is unlabeled because Nature is not among these labels. It's gray then, and we plot it behind all the colored points so that it doesn't interfere. But you see some gray here, like on top and on the bottom, and that's where the gray comes from. So we, even just looking at this picture, we see that there's a lot of structure here and a lot of structure that seems to make sense, right? So first of all, virology, all the papers on any given topic like virology or bioinformatics are close together. This makes sense. This means that our language model did a good job, and then our TSNE 2D visualization also did a good job. We see that things like psychiatry and psychology are close together and merge into each other, which also makes sense. We see that on the right, there's medical fields like cancer research in black here, or maybe cardiology, neurology, surgery in blue. Well, on the left part, it's more basic biology. And here in the bottom, there's a little bit of physics and computation and engineering, which is not supposed to be on PubMed, but somehow slips through, and PubMed still has some of it. Intentionally or not, I'm not sure. Moreover, we can now, so now we have this map, if you want, I will call it a map, and we can look at it in different ways. So we can color it by topic, as you see here. We can also color it by, for example, publication year, and then we see here all the papers are dark, and very modern, newly published papers are yellow, bright yellow. We see this very bright yellow cluster of papers published in 2021 and maybe 2020. And it turns out, if you look at what papers are in here, this is the research on COVID. So it makes sense that it's a sizable fraction of the entire literature, and it's all close together. So we call it a COVID island in here. And then one can zoom in and in different parts of this embedding and in various ways see that actually there's a lot of structure that is meaningful here on different levels. So there's more structure than you can see on this slide. Let me show it like that. We can zoom in into COVID just as an example. So here's this COVID island. I am zooming in. You see this shape of the COVID island, this yellow thing over here zoomed in on the right. And now I color it differently. Now I cover it not by journals, but I color, we assign topics in a different way based on the keywords in the titles of the papers actually, and coloring in a different way. And we see here that, for example, on top are more clinical COVID research, whereas on the bottom is social, mental health, anxiety, maybe the effect that pandemic had on the workers or students, and so on. So this is a relatively narrow subfield, right? A small part of this literature, but when we zoom in, we see more structure. And actually, there's probably even more if we were to zoom in on this little island, for example, over here. I have no idea what this island is, but I'm sure that this division into several sub-islands would still be meaningful. We looked at multiple examples like that. In the paper, we then use this embedding to look at different things. And I don't, this is not the focus of the talk. I want to come to paper mills. So I will just very, very briefly mention a couple of things. And if you're curious, you can later on look this in the preprint. I will not even list everything here that we have in the paper. So for example, just as an example, we can study things like uptake of machine learning methods in different parts of the biomedical literature. We were interested in that because we're interested in machine learning. So maybe I want to know where principal component analysis is used, or where support vector machines or deep learning is used. And then we can look for mentions of PCA in the abstracts. Then we can color this embedding by whether it uses PC, whether abstract mentions PCA or not. And then we see these islands that preferentially use PCA. And then we can go back here and see, okay, that was that field. And in that field, apparently, or perhaps like in the cardiology or in radiology, they like to use convolutional neural networks, things like that. That was interesting for us. But one can look at very different things. We can look, or we did look, it's in the paper, we could look at things like gender bias. We found it actually quite fascinating here. So we can extract from PubMed the author names, right? We can look at the first author, and we can look at the first name of the first author. Then using this, we can use some existing, essentially, lists of male and female names to guess the gender of the first author, which in many cases is impossible because maybe the name is not in these lists, maybe it's a rare name, maybe the lists are made based on the data from the United States of America, so a lot of Eastern names are not in there. So there's a lot of caveats in this analysis. Nevertheless, we get a high fraction of these genders estimated, and then we can see that there are more yellow areas which are more female dominated and more blue areas which are more male dominated. And it makes a lot of sense because this is the research, for example, on education. I would need to go back so that you see the labels again, but there's a lot of this more psychology was over here, psychology and psychiatry and education and nursery and things like that. And these are more female dominated areas, whereas surgery, for example, or engineering are more male. But what is interesting is that one can zoom in and look at like individual islands and ask, why is this is all blue? Because this is surgery, but there's this one little island, and this little island seems to be more female. There are more female authors there. Why is that? And then you look, and then you realize that actually this is the research on veterinary surgery. So it's surgery, but it's veterinary, and that's apparently a subfield where there's a high fraction of females. So the point of our paper is that this is a useful tool to study different things. And we advocate that one can use it for whatever, right? For whatever questions about the research biomedical literature as a whole, the corpus of biomedical literature that one can have. So one can do also silly things. Which fields like to say little is known in the abstract, or for the first time, or interestingly? It's not uniform. Apparently, there are clusters of research that like to say interestingly, and well, that was fun for us. That's one of the supplementary figures that we had. Then I'm coming to the point of the talk, is that we can also use the same map of biomedical research to study retractions. And that was originally not the intention at all. So we actually, in the first version of the preprint, didn't even do this. And when we put this online, we got comments about what if we look at the retracted papers. So we did the following. In PubMed itself, there are papers marked as retracted, as many of you will know. And then in addition, we looked at the retraction watch database, where more papers that are retracted are listed, because apparently some journals retract the papers but don't tell PubMed about that. So overall, we got something like 15,000 papers that are retracted in either of these two sources. And then we can look where they are. And we got this figure that is in the preprint as well. So we use two colors here, but that's actually not very important, I think. So black ones are flagged as retracted in PubMed. Red ones are flagged only in Retraction Watch, but not in PubMed. I think one can ignore that. Just look at where the retracted papers are here. So they are peppered all over this embedding, which makes sense. Like every now and then there is a retracted paper in any part, in any topic in biomedicine. Right? But there are clusters, and that's where it gets interesting. So there are clusters where this part has suspiciously many retractions, and this part has a lot of retractions over here. And just as a reminder, again, so that we're all on the same page, the embedding is only done based on the similarity between abstracts. There's nothing else that influences this embedding. So if there is a cluster here, like a little island, this means necessarily that all abstracts of these papers are very similar in each other, or at least the PubMed BERT model, the language model, believes that they are similar to each other. They have common words, they use common phrases, they are similar. And if there is a cluster, or let's say an island over here, that has a lot of retractions, this is strange, right? This means, or maybe this is not strange, but this could be a paper mill, or this could be something suspicious, because this means there's a lot of papers that are very, very similar, and a lot of them are retracted. But when I say a lot, overall, we have only 0.07% retracted papers in the entire literature. This is probably the most dense. Here's the fraction of retraction papers is the highest. So here's a zoom in with 4% of all papers are retracted. And if we take like just a tiny little island over here, there's almost 1/10, every 10th paper in here has already been retracted. And then in the paper, we say that if we look at this specific tiny little island that maybe only has 150 papers or so in it, then all of these papers are titled like microRNA something something does something else by targeting yet something else in osteosarcoma. All of these papers are like that. That's the titles. The titles were not taken into account, but the abstracts are also similar. That's why they form an island, and out of them, 11.8% have been retracted. The rest has not been retracted. So the question is, does it make sense that they are suspicious? They're very close to papers that have been retracted. Maybe all of them should be retracted, or maybe all of them should at least be inspected or something. When I wrote about that on Twitter, actually, there was some backlash also. So I'd like to discuss this today very much, because people were saying it's unfair to like scrutinize these papers. They didn't do anything wrong, perhaps. But we are now calling, we meaning the authors, like me and co-authors, are calling for additional scrutiny on these poor papers that just happen to be close to these papers that have been retracted. But I do think actually that this is very suspicious, and there is more than this figure shows. So I want to show some additional analysis that we did very, very recently that hopefully will make it into the final version of the paper, but it's currently not in the preprint. We just did it last week. And this was actually following Jennifer's lead, we talked briefly a couple of weeks ago, and she said, why don't we look at the countries where these papers are coming from? So PubMed, luckily, also contains information about the affiliations of the papers. So what we did, what Rita did, is to reprocess the entire dataset, extract the affiliation of the first author. In this case, we're only looking at the first author for simplicity. We look at we take affiliations of the first author. Sometimes there are multiple affiliations, as you all know. We take the first affiliation of the first author, and then we check which country name appears in that affiliation. And that's how we assign country to paper. Okay, so if it's international team, we may get something arbitrary and not representative of the paper, but in most cases, hopefully, it does represent where the paper actually come from. First affiliation of the first author. Okay, and now I'm coloring the retracted papers. So it's the same pictures here, just recolored. It's colored now by the country of origin. We see a lot of China, which should not be surprising for you, people studying paper mills. But we also see different colors here. This is, by the way, top 10 countries in the entire PubMed, not by retractions, but overall. And there's, like, we were looking at this just the other day, and there's so much curious stuff here. Like, for example, this is a German cluster, and this is a Japanese cluster. This is UK, I believe, and this little blue agglomeration is US cluster. Like, why is that? I'll come back to the Chinese clusters in a second. So we looked at it a little bit, and actually it was really fun because I looked at this. We looked at this. What is this? This is Germany, right? So we looked at Germany first because we are in Germany. Why is there a cluster of retracted German papers? Turns out all of them, or most of them, were authored by one guy, and they were co-authors, but he's on all these papers. Maybe some of you will know the name. It's an anesthesiologist who even has a Wikipedia article about how he has 200 retracted papers because he was caught in various kinds of research fraud. It's not a paper mill, it's just a specific person. It's a paper mill of one lab, if you want, and we can see it here. And I just today was looking at the several other clusters, and I realized that these blue papers over here coming from the States, United States, this is all work of Scott Rubin, whom I didn't know any of them, but actually they're famous. They have Wikipedia articles, and this green cluster is a Japanese guy working in bone research, and there's, like, you also find Science and Nature pieces covering each of these cases. So these are famous cases of research fraud. Okay, that's fun, but what is more interesting, I think, in the big scheme of things, are these enormous clusters that are not German, US, or UK colored. By the way, I didn't have time to check this United Kingdom cluster, so that still remains to be checked what this is. But this is China, and here in the zoom in, you see that all of these papers are from China, like everything. So we thought this is interesting, and we here only coloring retracted papers, but we can color all papers by the country of origin. Okay, and I'm coming to an end of my talk now. Here, all papers are colored by the country of first affiliation of the first author, whether it was retracted or not. So only a tiny fraction is retracted. I'm showing it here on the right. But if we look at where the biggest retracted clusters are, on top, these 3 islands on top, and here in the upper part. So this is here at the top, and this is this upper part. These are predominantly Chinese dominated areas. And actually, if one zooms in here, it's not even dominated by China. This is like 99 point something % Chinese cluster. Again, these are English abstracts. They are grouped just because they're similar abstracts, but all of them come from China. And a large fraction of them have been retracted here. So the question that I want to ask almost you, and I would like to discuss, is to what extent this basically leads to conclusion that this is all paper mill products or not. because we can see a lot of things here that maybe we can also discuss a bit later. Like, for example, there is a lot of magenta over here. Magenta is United Kingdom. So does this mean this is all paper mills from UK? I don't think so. Actually, I believe that the reason we see a UK cluster here is because UK has a different, Britain has a different spelling, right? They write behavior and analyze, not how they do it in the US. And we're analyzing texts. So I'm pretty sure that the reason we see this magenta slightly separate from the United States is just because of the difference in spelling. It's an artifact of that. And similarly, one can maybe explain away some other things that we see here, but it's really hard to explain away these islands that contain thousands and thousands and thousands of papers, all of them coming from one place, and this place is China. So I'm curious about the places in this embedding that have a lot of red, that basically are red islands, like over here, if you can see my cursor, here on the lower left, or maybe here on the upper right, and many other places that don't necessarily have a lot of retractions over here. Like, okay, here maybe there are some, but here not. So we just looked at it today. We didn't spend a lot of time, and I think we won't be able to do this for this publication, but this is an open question that I would really like to know the answer to. So I'm zooming in here in this little island over here, this double island. That's the zoom in. Okay, ignore the colors on the right. And I'm just looked at what these papers are. So here's just two examples. All of these and all of the other papers are super similar. So the papers in this island are all variations of the complete mitochondrial genome of something. And over here is the complete chloroplast genome of something else. Okay, almost none of that has been retracted. It's not, if you look at the number of retracted papers over here, this area is not suspicious at all. It is suspicious only because everything comes from China, or maybe it should not be suspicious. That is really an open question. So it could be a paper mill producing this kind of research, or it could be just that Chinese academia really likes to study these things. I don't know how to answer this question yet. Here's another example, an island on the top. I just zoomed in, and here are some titles that I won't read because I want to finish so that we have some time for discussion. But again, these are all very similar titles. They are all very similar abstracts. Basically, 100% of these papers come from China. So that's the open question. To what extent the homogeneous air, so the clusters in the text space that originate from a specific country can be an indicator of a paper mill activity. I'd like to close here with this open question. I will just mention that Rita just finished reprocessing the entire data set because, as I said, that was the data until 2021. Now we have data until the end of 2023, so we get 2 million abstracts more. We did everything, the same thing, again, from scratch. So the embedding looks a bit different, but overall, it's the same picture. There's actually quite a bit more red if you compare this picture on the right here with before, because apparently the fraction of papers that comes from China also increased in last years very strongly. So adding 2 more years of data actually increased the proportion, noticeably increased the proportion of red islands that one can see here. Okay, adding to my previous open question. And this will not be in the paper, but we have a website where you can look and explore this landscape interactively. So if you open this link, you see this embedding, the same one as in the paper, and you can zoom in with a mouse wheel as you do in Google Maps, and you can go to individual papers, you can click on it and brings you to PubMed. You can color things by you can open retracted papers over here and so on. So we plan, it's not done, but we plan to update the website to show the latest version eventually. And that's how it's going to be hopefully up to date also in the future. And that's how one can also ask questions like, what is this suspicious island? And any one of you can actually open this website already now and just look at what any of these little islands can possibly be. We don't have country information in there yet, but we will have it soon, I hope. Okay, with this, I would like to finish. Thank again. I realize that I don't have acknowledgement slide, so I just quickly go back so that it's in the video, and thank again all the co-authors. And I'm happy to discuss this and take questions.